objective, the precise fusion of two highly specialized haploid cells to form a completely new diploid organism. Achieving this union requires the flawless synchronization of two distinct anatomical pipelines operating inside two separate bodies. The male system operates as a continuous, temperature-regulated factory, generating millions of motile cells every day. The female system, by contrast, relies on a cyclical, hormonally gated environment that prepares exactly one cell per month. Following the trajectory of these cells reveals a precise sequence of events, from the independent genesis of the sperm and ovum through their physical collision to the 9-month cascade of embryogenesis. Every step in this sequence depends on exact timing. An offset in the endocrine clock or a structural blockage in the ductal pathways prevents the initiation of new life. The primary physical constraint of male reproduction is thermal. The synthesis of viable male gametes requires an environment roughly 2 to 2.5 degrees Celsius cooler than the human core body temperature. The body solves this thermal requirement by housing the testicles externally in the scrotum, keeping the internal temperature strictly regulated below the body's baseline. Inside this cross-section of the testicle, the tissue is partitioned into roughly 250 testicular lobules. Each lobule is packed with highly coiled structures called seminiferous tubules. The inner lining of these tubes holds diploid male germ cells known as spermatogonia, embedded alongside larger supporting cells. This is where spermatogenesis occurs. The germ cells undergo continuous meiotic division. A diploid spermatogonium splits, leaving one half at the tubule edge to maintain the population, while the other divides to form 4 distinct haploid spermatids that move toward the hollow center. This entire assembly line activates at puberty, when the hypothalamus releases gonadotropin-releasing hormone, stimulating the anterior pituitary gland to secrete luteinizing hormone and follicle-stimulating hormone. These two hormones strike dual targets. Luteinizing hormone acts on Leydig cells outside the tubules, forcing them to synthesize androgens. Simultaneously, follicle-stimulating hormone triggers the Sertoli cells within the tubule to physically nourish the rapidly dividing germ cells. Newly formed sperm cells are initially non-motile. They are swept through a network of ducts called the rete testis and deposited into the epididymis, a coiled tube along the back of the testicle where the sperm are stored and eventually acquire motility. During ejaculation, sperm are propelled upward through the vas deferens. Along the way, they mix with fructose-rich fluids from the seminal vesicles and calcium-heavy secretions from the prostate gland to form semen. The male system operates as an industrial pipeline, built for the continuous, high-volume production and chemical armoring of millions of microscopic delivery vehicles. The female timeline operates entirely differently. Oogenesis begins before birth, while the female is still a fetus, and pauses completely until she reaches puberty. This dual-axis display tracks the female biological countdown. At the onset of puberty, the ovaries hold hundreds of thousands of dormant primary oocytes trapped inside protective casings known as primordial follicles. During the first 14 days, the follicular phase, slowly rising levels of follicle-stimulating hormone and luteinizing hormone force a primary follicle to mature. It rapidly expands, building a thick outer layer of granulosa cells and a fluid-filled cavity called an antrum. As this follicle grows, its granulosa cells secrete increasing amounts of estrogen. This rise in estrogen triggers the proliferation of the endometrial lining, thickening the uterine wall in preparation for a potential embryo. Rising estrogen levels trigger a critical threshold, prompting the pituitary gland to release a massive upward spike in luteinizing hormone. This chemical surge ruptures the mature follicle, ejecting the oocyte out of the ovary. This ejected secondary oocyte is immediately collected by the fimbriae of the fallopian tube, where it begins drifting inward, awaiting fertilization. Back in the ovary, the luteal phase begins. The ruptured shell of the follicle collapses and reorganizes into a dense yellow structure called the corpus luteum. The corpus luteum takes over endocrine control, pumping massive amounts of progesterone into the bloodstream. This hormone maintains the heavily thickened, vascularized uterine lining, ensuring it remains receptive to a fertilized egg. If no fertilized egg arrives to embed itself, the corpus luteum simply degenerates. Progesterone levels plummet, the structural support vanishes, and the uterine lining collapses and is expelled as menstruation. The female reproductive cycle is an uncompromising biological countdown, repeatedly constructing and destroying a highly specialized environment based strictly on the rise and fall of hormones. Convergence requires millions of motile sperm to navigate through the cervix, cross the uterus, and enter the fallopian tube to reach the ampullary isthmic junction. They are searching for the drifting secondary oocyte, which is heavily armored by an outer cellular barrier called the corona radiata and a tough inner glycoprotein shell known as the zona pellucida. Fertilization demands physical penetration. The sperm bombard the outer barrier, initiating the acrosome reaction. An enzyme-filled cap on the sperm's head breaks open, chemically dissolving a path right through the thick zona pellucida. The instant a single sperm breaches the membrane, it triggers the cortical reaction. The entire zona pellucida chemically hardens in milliseconds, permanently locking out the thousands of other sperm competing for entry. This penetration physically forces the oocyte to complete its final meiotic division, leaving behind a secondary polar body and transforming into a full mature haploid ovum ready to merge nuclei. The genetic sex of the resulting zygote is decided in this exact millisecond. If the successful sperm carries an X chromosome, the pairing dictates a female. If it carries a Y chromosome, the pairing dictates a male. This fusion instantly terminates the female system's cyclical countdown and shifts the body's internal state entirely to gestational maintenance. The newly formed diploid zygote does not stay in the fallopian tube. It begins a multi-day descent toward the uterus. As it drifts, the zygote undergoes cleavage, a series of rapid mitotic cell divisions. It splits from 2 cells into 4, 8, and 16, increasing the cell count without actually growing in total mass. This dividing mass transforms into a blastocyst. The solid ball of cells hollows out and strictly segregates into 2 distinct cellular populations. The outer layer is the trophoblast, biologically programmed to expand outward and penetrate the surrounding maternal tissue. Grouped together on the inside wall is the inner cell mass. This dense cluster of stem cells possesses the ability to eventually construct every physical organ in the human fetus. Once the blastocyst reaches the uterus, the trophoblast chemically drills into the thickened endometrium. In response, maternal uterine cells divide rapidly to surround the blastocyst, completely burying it within the uterine wall. Implantation marks the structural initiation of pregnancy. A free-floating cluster of cells successfully integrates into the maternal tissue, establishing the direct vascular link required for sustained growth. Following implantation, the inner cell mass differentiates into 3 specific germ layers. The ectoderm maps out the nervous system and skin, the mesoderm forms the heart and muscles, and the endoderm shapes the entire digestive tract. Simultaneously, the trophoblast extends finger-like projections called chorionic villi that interlock directly with uterine tissues, assembling the placenta. This new structural organ executes a chemical takeover. The placenta pumps human chorionic gonadotropin, human placental lactogen, and massive amounts of estrogen and progesterone into the mother's bloodstream, definitively blocking the menstrual cycle from restarting. By the end of the 9-month gestation period, the fully formed fetus achieves maximum physical capacity within the confines of the uterus, shifting its position in preparation for delivery. Delivery is initiated by the fetal ejection reflex. The mature fetus and the placenta secrete specific hormones that trigger the first mild contractions of the uterine muscles. As shown here, these initial contractions alert the maternal pituitary gland to release oxytocin. This triggers a strict positive feedback loop. Contractions force the brain to release oxytocin. Oxytocin acts on the uterine muscles to physically force stronger contractions. Stronger contractions, in turn, demand the release of even more oxytocin. This self-reinforcing chemical loop guarantees stronger contractions with every passing minute, inevitably culminating in parturition, the mechanical expulsion of the baby, followed quickly by the placenta. Birth is an explosive, mechanically forced eviction, driven entirely by an unbreakable hormonal chain reaction inside the mother's body. Human reproduction relies on this biological domino effect, stretching sequentially from the microscopic mechanics of meiosis to the macroscopic physical force of childbirth. The entire pipeline depends on extreme precision. A single degree of elevated temperature in the testes, or a missing hormonal surge in the pituitary gland, breaks the sequence instantly. This system represents a precise biological mechanism, one that systematically re-engineers a human body for the singular purpose of constructing another.